Light energy is used to make ATP and NADPH. Photosystem 1 and 2 reaction center chlorophylls P700 and P680. But what happens to the light energy that is absorbed? We don't see plants live glowing like light bulbs, but we also know that energy can't just disappear. Light absorption in PS2 when light is absorbed by one of many pigments in photosystem 2. Energy is passed inward from pigment to pigment until it reaches the reaction center. ATP synthesis, the high-energy electron travels down an electron transport chain, losing energy as it goes. Light absorption in PS1, the electron arrives at photosystem 1 and joins the P700 special pair of chlorophylls in the reaction center. When light energy is absorbed by pigments and passed inward to the reaction center, the electron inside P700 is boosted to a very high energy level and transferred to an acceptor molecule. NADPH formation. The high energy electron travels down a short second leg of the electron transport chain. At the end of the chain, the electron is passed to the NADP+, along with a second electron from the same pathway to make NADPH. The net effect of these steps is to convert light energy to chemical energy in the form of the ATP and NADPH. What is photosystem? Photosystem 1 versus photosystem 2. Special pairs. The chlorophyll A special pairs of two photosystems absorb different wavelengths of light. The PS2 special pair absorbs best of 700 nm. Because of this, the special pairs are called P680 and P700 respectively. Primary acceptor. The special pair of each photosystem passes electron to a different primary acceptor. The primary electron acceptors of PS2 is PLPT, an organic molecule that resembles chlorophyll, while the primary acceptor of PS1 is a chlorophyll called A. Source of electrons. Once an electron loss, each photosystem is replenished by electrons from different sources. The PS2 reaction center gets electrons from water, while the PS1 reaction center is replenished by electrons that flow down an electron transport chain from PS2. During the light-dependent reactions, an electron that is excited in PS2 is passed down an electron transport chain to PS1, losing energy along the way. In PS1, the electron is excited again and passed down to the second leg of the electron transport chain to a final electron acceptor. When the P680 special pair of photosystem 2 absorbs energy, it enters an excited high-energy state. Excited P680 is a good electron donor and can transfer its excited electron to the primary electron acceptor, PLPT. The electron will be passed on through the first leg of the photosynthetic electron transport chain in a series of redox or electron transfer reaction. After the special pair gives up its electron, it has a positive charge and needs a new electron. This electron is provided through the splitting of water molecules, a process carried out a portion of PS2 called the manganese center. The positively charged P680 can pull electrons off of water, which doesn't give them up easily because it's extremely electron hungry. When manganese center splits water molecules, it binds two at once, extracting 4 electrons, releasing 4 H+ ions, and producing a molecule of O2. About 10% of the oxygen is used by mitochondria in the leaf to support oxidative phosphorylation. The remainder escapes to the atmosphere where it's used by aerobic organisms such as us to support respiration. When an electron leaves PS2, it is transferred first to a small organic molecule, plasticomat, then to a cytochrome complex, and finally to a copper-containing protein called plastocyan. As the electron moves through this electron transfer chain, it goes from a higher to a lower energy level, releasing energy. Some of the energy is used to pump proton from the stroma into the thylakoid interior. This transfer of H plus along with the release of H plus from the splitting of water forms a proton gradient that will be used to make ATP as well used shortly. Once an electron has gone down the first leg of the electron transfer chain, it arrives at P(S1), where it joins the chlorophyll, a special pair called P700 because electrons have lost energy prior to their arrival at P(S1). They must be re-energized through absorption of another proton. Excited P700 is a very good electron donor. It sends its electron down to a short electron transport chain. In this series of reactions, the electron is first passed to a protein called ferredoxin, that is transferred to an enzyme called NADP+, to make NADPH. NADPH will travel to the Calvin cycle where its electrons are used to build sugars from carbon dioxide. After leaving P(S1), cyclically flowing electrons travel back to the cytochrome complex or plasticanol in the first leg of the electron transport chain. The electrons then flow down the chair to P(S1) as usual, driving proton pumping and the production of ATP. The cyclic pathway does not make NADPH since electrons are moved away from NADP+ reductive. At least in some cases, chloroplasts seem to switch from linear to cyclic electron flow when the ratio of NADPH to NADP+ is too high. In addition, cyclic electron flow may be common in photosynthetic cell types with especially high ATP needs. Too little NADP+ is available to accept electrons. Synthesizing bundle sheet cells of plants that carry out C4 photosynthesis. Finally, cyclic electrons flow may play a photoprotective role, preventing excess light from damaging photosystem protein and promoting repair of light, including damage. How long do she think